All right, so our next speaker is Jay Davis, and he works at the San Francisco Estuary Institute, and he is also the principal investigator for the Bioassessment Oversight Group, Bioaccumulation, thank you. I always do that. I'm new to this game, sorry. So he's a strong partner in SWAMP, but he also serves as the lead investigator for the regional monitoring program in the San Francisco Bay Estuary. So, and Jay has his PhD from UC Davis. I'm noticing a trend of UC Davis. But that's all right. Thanks, Jay.
Thanks, Greg. Go Aggies. It's great to be here today for this first Water Quality Science Symposium. I think it's a great idea and hope that this is the beginning of something that continues for a long time. I thought I would start with some general information, defining what bioaccumulation is, because even sometimes the scientists kind of get that a little wrong. And then I'd like to give a general overview of bioaccumulation in California and kind of the big picture on what the concerns are. And I'm going to do that in the form of a preliminary report card that's just my own creation. It's not an official SWAMP product, but just something to kind of provide fodder for discussion. And then I'm going to summarize what SWAMP is doing on this issue. So first, bioaccumulation is a general term that describes the uptake of toxic chemicals by any living thing, any biological organism. And it occurs at different levels in the food chain. I'm showing a diagram here that kind of shows the lower trophic levels leading up to the higher trophic levels. The major points of concern from a water quality management perspective are the exposure of humans that eat fish and shellfish. And the fish and shellfish accumulate contaminants either through transfer through the food chain or directly from water. And then there's also significant concern for effects of contaminants on wildlife. So especially highly exposed are species at the top of the food chain like marine mammals that eat fish and fish-eating birds. A particular kind of bioaccumulation that's very important in kind of determining the level of risk and concern is biomagnification. And this is a subset of the broader category of bioaccumulation where the concentrations in the food chain increase with each step that you go up. So this figure is showing the mercury cycle, and you'll get a clear idea of why I'm focusing on mercury here as I go through my slides. It's the major concern for bioaccumulation in the state. And mercury, as an example of biomagnification, works its way up through the food chain, starting in sediment, primarily in anoxic areas in sediment where mercury is converted to methylmercury. And methylmercury is the form that is highly bioaccumulative and highly toxic. It's really rapidly accumulated by phytoplankton, very strongly accumulated by phytoplankton. And then the consumers of the phytoplankton are a little bit higher in their mercury concentrations. Small fish that eat these little invertebrates are higher still. And then the piscivorous species that eat the small fish receive an even higher exposure. And then the highest exposure can be for people at the very top of the food chain. So that's the quick intro on bioaccumulation. So now on to the report card. And as you can tell from the cartoon, it's a mixed bag. So there's some good news and some bad news. So I'm going to present the summary based on a framework that we developed for the Bay Regional Monitoring Program for emerging contaminants. But I think it's a useful framework for looking at contaminants more generally. And I'm going to place things into these 4 tiers. The first tier is contaminants that are of high concern, And these are contaminants where there are significant impacts on beneficial uses. There's significant threats to public health or wildlife health. And this results in closures of fisheries or consumption advisories or actions to try to reduce exposure of humans and wildlife. So that's the highest tier. Tier 2 are contaminants of moderate concern where there's some evidence of impact but not high enough to reach the level of sort of regional or statewide actions in terms of closures or TMDLs. But you know, it's either more localized or less clear that it's a really significant exposure and impact. Tier 3 are things that are of low concern, and many contaminants fit into this category where we don't, you know, the concentrations that we see don't approach thresholds, and you know, we can't say that they're of no concern necessarily, but based on the information we have, they're of low concern. And then there are also many chemicals that fall into this last category of unknown concern, where there's a lot of new chemicals that, new pesticides, many other chemicals that are used in commerce that come onto the market where we don't have data from the environment and we don't know how toxic they are. Emerging contaminants fall into this category. So those are the 4 categories. First I'm going to focus on contaminants and the fishing beneficial use. And at the top of the list here is methylmercury. The map shows data from 2007 to 2011. We had a 5-year period where SWAMP collected data on a statewide basis on contaminant accumulation in sport fish. And the red dots show locations where the concentrations were of significant concern. The green dots show locations where concentrations were below levels of concern, and then orange and yellow were in the middle. I really want you to focus on the red and the green. But before we began these statewide surveys, there had been monitoring done across the state, but it wasn't done in a systematic manner, so we really were filling in a lot of uncharted territory, and some areas had been sampled. Other areas hadn't really been sampled at all. So in this period, SWAMP has really filled in the map of our understanding of mercury bioaccumulation in the state. Particular areas of concern are warm water habitats in the Central Valley and foothills, where we typically see largemouth bass, and we like to monitor largemouth bass, and I'll show why. And then also on the coast, we were kind of surprised to see consistently high concentrations of mercury up and down the coast. This was an area that hadn't really been sampled thoroughly before, but we see species out there that are long-lived, and mercury accumulates over the lifespan of the fish, so we see high concentrations there too. Those are the 2 regions of particular concern. The good news is in higher elevation, colder water lakes that are dominated by trout, we typically see low concentrations. And thinking broadly, we'll be hearing more about HABs and algal toxins later today, and this isn't something that we're monitoring in the SWAMP bioaccumulation monitoring program, but SWAMP is looking at these chemicals through the HAB work. But algal toxins like saxitoxin, domoic acid, routinely cause fishery closures, shellfish closures annually on the coast. And then last year, I'm sure everybody heard about the Dungeness crab closure that was due to domoic acid accumulation. So again, major impact of fishery closure, they fall into this high concern category. Into the moderate category, PCBs are the contaminant of second highest concern that we routinely monitor in SWAMP. And the map here shows a lot of green, and then there's a fair amount of yellow. There's only a few red dots that are the areas where the concentrations are high enough where advisories might say not to consume fish in these areas. San Francisco Bay is one of them, San Diego Bay, and then there's a few other, there's a couple reservoirs in Southern California. So some areas, this is kind of a more localized phenomenon where we see high concentrations. And aside from that, levels of moderate or low concern, but overall it fits into the moderate concern category. Microcystin is another algal toxin that in the Klamath Basin is a concern accumulating in freshwater mussels. So putting this in the moderate concern category. And then on the good news side, we have measured a lot of things, both in swamp and in other programs, that fall below thresholds of concern. And the map here is showing selenium as an example from a human health perspective. We've looked at this quite thoroughly, and in almost all cases, selenium is below all levels of concern for human exposure. And then there are some contaminants where we don't have great information. I mentioned emerging contaminants, CECs. Perfluorinated chemicals are a category of chemical that we've been monitoring recently where we don't really have enough information. Dioxins, we have some information from San Francisco Bay, but not elsewhere. On aquatic life, again, methyl mercury is at the top of the list of contaminants of concern. In San Francisco Bay and across the state, we see concentrations in birds that are high enough to affect survival of embryos. And this is thought to be a significant risk to these populations. So those are the two most extreme cases, but then there's also concern for ridgeways rails in San Francisco Bay, which is an endangered species. Marine mammals get high exposure through their fish diets. There's even concern for the fish themselves, like largemouth bass, because at the levels we see in their tissue, they could be experiencing problems from mercury exposure. Brian mentioned earlier the hepatotoxicity, liver toxicity of microcystin, and this is another case where there's concern for endangered species where microcystin, which was generated in a freshwater environment upstream of Monterey Bay, was transported downstream into Monterey Bay and caused sea otters to die. So this is a kind of a rare case where there's actual, clear mortality due to a contaminant. And here's the otter, and this is the picture showing symptoms of liver toxicity. There are also other biotoxins. In 2011, there was a big abalone and shellfish die-off on the Sonoma County coast. So again, algal toxins are a major concern out there. In the moderate concern category for aquatic life, selenium is a concern in San Francisco Bay. There was a TMDL just established for selenium in North San Francisco Bay. There's also a TMDL for selenium in Newport Bay. The primary concern in San Francisco Bay are white sturgeon. And again, the early life stages of this species are at risk from the selenium exposure that they're seeing. A recent study found that an old bad actor, DDT, which wasn't really wouldn't have made my list until I saw this study a few years ago, but DDT is still out there in certain cases, certain places at high enough concentrations to reduce eggshell thickness and reduce reproduction of California condors because they're feeding on marine mammals at the very high in the food chain, and these reintroduced condors experience these DDT symptoms. PCBs are also a concern in fish-eating birds and marine mammals. Again, there are a lot of things that we measure that are below levels of concern, so it's not all bad news. and then there are more unknowns. So this is the overall summary of what we're really concerned about, things in the top two tiers, mercury in particular in terms of the anthropogenic chemicals that my part of the SWAMP program is focusing on. Mercury is the number one concern. PCBs are number two, and then selenium, DDT are also of some concern. So now on to what SWAMP has been doing to address this issue. And this work is done under the guidance of the Bioaccumulation Oversight Group, the BOG, and then also through under the guidance of the SWAMP Roundtable and SWAMP management in general. And I'm going to break it down by water body type. We cover all the major water body types where fishing is a major beneficial use. So we've focused a lot of our attention on lakes. That first map I showed, in that 5-year period, we covered over 400 locations, and 272 of the locations were lakes. So in 2007 and 8, we sampled 272 lakes. And then we moved on to the other water body types for 3 years. In 2012 and 13, we came back to lakes to look at exposure of wildlife and did a study looking at grebes on lakes and characterizing their exposure. So that was 2012-13. And then 2014, 15, and 16, we've done other lake studies. In 2014, we focused on the cleaner end of the spectrum and tried to validate that some of the lakes that we saw low concentrations in really did have low concentrations, and this is kind of the other side of the coin. There is some good news, even for the worst contaminant, mercury. There are water bodies where concentrations are low, and even in the contaminated water bodies, there are species that are low. So it's important to understand the positive side of the picture as well. In 2015, we started a long-term program for monitoring mercury in largemouth bass lakes. This was one of the primary regions of concern that I mentioned earlier, and I'll show a picture about that. And then in 2016, we've done some additional, we're doing some additional lake sampling to sample some unsampled lakes and to fill some information gaps. There have been some significant outcomes from the work that we've been doing. There's been a lot of 303D listing. Hundreds of lakes have been added to the 303D list of impaired waters, which kind of brings them into the regulatory fold and requires the water boards and permit holders to do something about the contamination. One of the forms of action is statewide TMDL that's in development for reservoirs. The first statewide TMDL for California. Also, this work resulted in the first statewide consumption advice for lakes from the Office of Environmental Health Hazard Assessment. And then there's been a lot of updates of specific consumption advisories for water bodies as well. One of the highlights of the data set that we've generated are water bodies where we've sampled largemouth bass. Largemouth bass have proven to be a really great indicator. I'm showing a litmus paper photo here. They're a great litmus test for mercury contamination in water bodies. Mercury, because it accumulates over the lifespan of an organism, how big the fish that you sample is really important in determining the concentration. So it's really important to adjust what you measure for the size, to take that size into account. So we have a technique where we can estimate the concentration for a 350 millimeter bass, and that's the index that we use to really compare lakes. We've done this at 157 lakes to date, and this is the data set. Not expecting you to read all these lake names here, but they're all in order, and the concentrations range from well over a part per million to over a couple orders of magnitude to below around 0.01 parts per million. And some of the key lines to compare to, 1.31 parts per million is the no consumption threshold for the less sensitive part of the human population, men and women beyond childbearing age. A small percentage of lakes, only 2 lakes were above this level, but this threshold of 0.44 parts per million is for the sensitive population, which is children and women of childbearing age. And above this level, OEHA starts to think about issuing a no consumption advisory. 30% of the lakes had bass that were above this level. And then 0.22 parts per million is the low end for the non-sensitive population. And 35% of the lakes were below that level, 65% were above. So 35% were basically below levels of concern for the non-sensitive population. For the sensitive population, the lower end is 0.07, so it's a pretty small percentage that were below the lowest level for that. I mentioned that we've got a long-term plan for monitoring these bass lakes, this important category of water body. And we've identified about 190 of them, and we've come up with a plan for providing updated information for each lake on a 10-year cycle so that we can track the status of these water bodies. And we came up with a way of doing this, which will allow us to develop, each time we go out, and to fit things within the budget, we do 5 rounds over the 10 years. So every odd year we go out and we sample 1/5 of these lakes. And each time we do it in a randomized way. So we're getting, each year we'll get a good estimate of the statewide average for these lakes. And then over time we'll be able to track trends in the statewide average, which is a big data gap. Currently, we don't really have any good information on regional trends in mercury. And this is important to get, not only because there are control actions happening in the state through TMDLs that are not at the state. Well, there's the state reservoir TMDL, which when that kicks in, we would expect, hope for that to reduce concentrations. But there are also global forces that can reasonably be expected to influence mercury, climate change, changing emissions from Asia. Emissions have been increasing in Asia, but hopefully they're going to start tailing off soon. These things can cause statewide changes in mercury that it's important to understand so that we really can understand the impact of the management actions that we take. I mentioned the wildlife study, and this map shows the data from that, where this effort focused just on mercury because of the risk to fish-eating birds. And this is the western grebe, which was western grebes and Clark's grebes, closely related species, were what we sampled. We were able to do this at 25 lakes, and again, you see some red dots popping up. About 7 of the 25 lakes had concentrations that where 50% of the individuals sampled were at moderate risk of impairment. So there's significant risk at many lakes, and we also developed monitoring tools here by looking at prey fish and sport fish along with the birds. On the coast, we sampled in 2009 and 10, and we coordinated with the Bay Regional Monitoring Program and the Southern California BITE program, and really filled in with good, thorough sampling of the coast. And again, this is an area where we saw consistently high mercury concentrations. Coming very soon is, as a result of this work, is statewide consumption advice for the coast from OEHA. And rivers and streams, we did a 1-year sampling effort in 2011. And as for the lakes, this is providing support for the 303D listing and TMDLs in the form of, particularly in the form of implementation of the delta methylmercury TMDL. So along the way, we've produced reports, fact sheets. We've put our data into CDN, and this is a picture showing the different fact sheets that we've generated. These are all available on the SWAMP website. I'm going to show how to get there at the end. I forgot I had the spinning. This topic is of high public interest. People really care about their health, and whenever we produce a report, the media pick up on it. And this is just a couple of examples from recent work. And because of that, understanding that, and also understanding that mercury is a contaminant that the roots of the contamination go back to the gold rush, and it's a difficult task to try to reduce levels in the environment, but something that we can do in the meantime is inform people about the patterns in terms of what water bodies are high and low and what species are high and low. So we've put a lot of effort into the Safe to Eat portal component of the My Water Quality website, and I'm showing a screenshot of it here, where all of the data that I'm showing are accessible. Users can generate the same kinds of maps I've been showing, and we're about to produce a revised, updated version of this, but we put a lot of effort into making these data available to the public. So, wrapping up, for more information, the easiest way to get to the particular content I've been showing on the SWAMP website is to just Google Bioaccumulation Oversight Group, and you can find all of the documentation there. If you'd like to be on our email list, you can email me, and I can arrange for that. And then the My Water Quality portal is a great way to access the data as I showed. That's it.
We have time for a couple of questions.
First off, thank you for your presentation. It was very helpful. About a third of my work is having to do with mercury. But I was just curious, out of all of those lakes, what was your, did you have a minimum amount of largemouth bass you had to catch for each lake?
Well, in order to get that length adjusted concentration, we basically do an analysis of covariance. So we try to get a dozen fish from small size to large, and then basically do a regression, and then we can estimate the concentration at that particular size. So we do that across all the lakes, and that gives us that good litmus test. If we didn't do that, some lakes you get large fish. There's a very steep slope to that relationship of size and mercury. So if you get large fish, they're going to be high, even from a relatively clean lake, and if you get small fish, they're going to be low. So that's how we account for that factor. So basically 12, and then if it's a larger lake, then we do multiple locations.
Hi. Are you able to use the monitoring data to forecast? Have you tried doing that?
We haven't put a lot of effort into trying to develop models yet. I think the key thing to do to head in that direction is to start getting some trend information. I mentioned that Clean Lakes effort, and it was the first time where we did some revisiting of sites. And it was kind of surprising how consistent the concentrations were over about a 6-year period from, we did the initial work in 2007 and 8, and then revisited some of them in 2014. And I was surprised at how consistent they were across that period. So in order to, we need trend information, and one of the important things types of information to gather is how much variation there is from year to year. We don't really have a good idea of what that looks like. But, that's kind of the next step. But also we are thinking about how we could use the data we have to develop predictive models. We can use the spatial information we have to try to understand what's driving mercury, and develop models that way. And maybe it'd be a bit of a stretch without having any trend information, but we could use that type of modeling to do some prediction. Yeah.
I just had a question thinking about those tier 4 chemicals, kind of the unknown concern. What's the process, or how quickly do chemicals get added to the analyte list? To maybe expand. And also, do you archive your samples for future analysis?
Yes, we do archive, and we with that particularly emerging contaminants in mind. As far as the what we've been doing in this with the swamp monitoring, we've been coordinating with major regional programs like the Bay RMP and the BITE program. With the funding we've had, we've really been trying to cover as many water bodies as possible, and looking at emerging contaminants, they're expensive to analyze, so it has a big budget impact. So we've been focusing, in the initial surveys, we had a broader suite with mercury, PCBs, legacy pesticides. We did a little bit of PBDE work, but we've been kind of narrowing our focus recently, and we're really relying on the other programs that have more resources and are active on emerging contaminants to kind of be the pilot areas for CEC work. And the Bay RMP, which I'm also involved with, does a ton of work and looks really closely at that. And I think that's I think that's a good approach, because places like San Francisco Bay and the Southern California BITE are sort of the worst case scenarios for CECs. And so I think it's good to have them sort of out front, screening things, and then after they identify things that, like the RMP uses that same tiering system, once things kind of start climbing in their tiers, then it makes sense to think about it for statewide monitoring.
Thank you very much, Jay.